Patient, I'm at work. Can you see my new background? So hopefully I don't know if I've got rubbish Wi-Fi. Hopefully my Wi-Fi is okay. Hopefully you can all hear and see me okay. And my rubbish uni internet doesn't drop out. So let me know in the chat if you can hear me and see me, because today we are doing big catch up. First lesson, we're going to do biology. So today we're doing intro to cells. I am Hannah. Nice to meet you all. If you're new, welcome. Just say hello in the chat. How many people have we got? I've got 2 laptops on the go today. So I've got you guys on this laptop and I've got my lesson on this laptop. Okay. So while we are waiting for everybody to arrive, instead of a fun fact for the start of this week, I just want to give you a couple of tips about A-levels and what I found really useful doing A-levels. Hello everyone. Hello Catherine, Laura, Talitha, Olive, Grace Stingray. Hello everyone. So I'm just going to pop you on my screen share. Whoop. Okay, so if you are at this lesson today, here we've got you. I'm going to put my laser pointer on. This should be you. You should have finished your GCSE biology, and future you should be possibly starting your A-level biology or A-level human biology. When I did my A-levels, we did human biology. So this is like kind of aimed at age 15 to 16 plus. Hi Miss 91, hi everyone. So you should be going into A-level biology come September. If you're not and you just bloody love cells, then that's okay. You're more than welcome to join the lesson. But this will be aimed at ages 15 to 16 plus. So before we start, this is probably my number 1 tip. Sentence book, it's going to be a long day. It's a big day of lessons today. Olive, if you're not 15 yet, don't worry, you can still come to the lesson. So my biggest tip for A-level is use the mark scheme. So whatever exam board you're part of, when you start your A-levels, they'll tell you you'll be AQA, OCR or EdXL. So these are the people that run the exam, that make the exam. And if you Google, for example, EdXL mark scheme biology, it will come up with something like this. And it will say things like, you know, the student must know all of these bits about cells. So here, you must know the functions of eukaryotic cells and you must know all of these things. So when you're like revising for your exams, this is what I did. I would go through the mark scheme and I would literally like tick off everything. I'd be like, yes, I can do that. Yes, I can do that. Then when you get to the exam, you like physically cannot be surprised about the questions they're going to ask you because you've already done everything the exam board could possibly ask you to do. So this is literally like the exam telling you, we would like you to know all these things for the exam. So this is what I used to use for organising all my revision. And I'd be like, yeah, bam, 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 tick, tick, tick. And I'd know it all, right? Oh, sorry, just text messages. So this was my recommendation for revising for your A-levels, is use the mark scheme to direct your revision, because then you know exactly what you need to know for the exam and you cannot be surprised, right? So your lateness is excused, Leonardo, don't worry. Right, so before we get started on cells, we need to know what makes a living thing a living thing. And when I learnt this, it was Mrs. Gren or Mr. Nug. But at the moment, this acronym, Mr. Sheng, these are going to be all the characteristics that make up a living organism. So to be alive, you must do the following things, right? So I'm going to give you the first one to start you off. Zenon has put movement. Whoa, movement is not one, my friend. Right, I'm going to give you one, and I'm going to go metabolism. Okay, what do we think, what is metabolism? If I am metabolizing something, what am I doing? So this is the first M, metabolism. Zenon's put, oh, this stuff. Someone's put respiration. Zenon, I don't know this stuff. That's okay. Right, number 1 question. What is metabolism? How would you define metabolism? and I'm going to put a little text box instead. So Karina's put burning food. I'll give you half a mark, yeah? Laura's put reactions are happening in organism. I will give you half a mark, we're halfway there. So we're not going to go into too much detail about metabolism because it is an enormous topic. Ollie's put changing molecules, yeah? So we're going to put essential chemical reactions that happen in the body, right? So these are just chemical reactions in the body that in the end will generate energy. But we're just going to call them essential chemical reactions for life. Oh, Ms. Greensaver just went on. Right. Second R, what do we think this R is? So if you are alive, who's taken a mick out of my spelling, Leonardo? Metabolism. Right, so what do we think the second R is? I'll give you a clue, it's about having children. So the second R has to be, I'll give you the second letter. What is the second R for splitting me into two people? So we're going to go reproduction, okay? I can't say it. Right, reproduction. This is the second thing. So living things can produce offspring, and this can be sexually or asexually, okay? This is what bacteria do. This is what we do, okay? So we're just making offspring via these two processes. Asexual reproduction, which is bacteria normally, and sexual reproduction, which is us guys, okay? Then we've got S. What do we reckon the S is? If I am alive, I must do something beginning with S. Zenon's put sensor. So what do we think the S is? If I am alive and I must do something beginning with S. And it's changed over the last couple of years with A levels. Someone's put speech. Olive, I'm going to give Olive the point. It's sensitivity. Sensitivity. And by sensitivity, I just mean organism can respond. We'll put it in text. So I respond to external and we'll put internal stimuli. So external stimuli might be a plant and the sunlight shines on the plant and then the sun is like moving to the plant to get them more sun. Or it could be us responding to things like temperature or things like, so temperature, or if I'm thirsty, or all sorts of things. So I can respond to lots of different stimuli. Sensitivity is S. And what do we think the H is? I am alive, I must do something beginning with H. This must be oh. I'll give you the second letter. Oh. What must I do beginning with H if I am alive? I'll give you the third. Home. Someone in the chat. Catherine's put hormones. That's a good suggestion. Heart. Another good suggestion. Homeostasis. My favourite word. Yes. So homeostasis is all about maintaining a stable internal environment, right? I know that sounds like a bit of a annoying thing. So what I want to do is I'm maintaining a stable environment. And that means things like my core body temperature is 37 degrees about, yeah? So I'm always trying to maintain my body temperature around 37 degrees. Basically, homeostasis means I don't want extremes of things in my body. So I don't want to be really boiling hot, then really boiling cold, right? I don't want my heart rate to be really fast and really slow. I want everything to be nice and chill, nice and stable all the time, okay? That's what homeostasis is. What about E? If I am alive and I need to do something beginning with E, what do you reckon E is? Oh, Olive's already put excretion, yes. So we're pooping, excretion. Okay, so I am removing my waste products. Removal of waste. Okay, that's all excretion is. Oh, Catherine, did I say boiling cold? I meant boiling hot and freezing cold. So excretion's removal of waste. What about N? What do we think N is? If I am alive, I must do something beginning with N. N must be nutrition. Yeah, you guys have got it. Let me just stop my screensaver from going off. Nutrition. So I must be able to exchange materials and gases with the external environment. So for plants, obviously it's not the same nutrition as us. And what do you reckon the G is? The last one for G, if I am alive and I must do something beginning with G, it is growth. Yes, correct, Olive. Right. So in the exam, you can see this is going to be a nice 1, 2, 3, 4, 5, 6, 7-mark question. Yeah. So it will be in the exam for 7 marks. Please give the 7 characteristics of all living things. And you can be like, Mr. Sheng. And now you know all of them. So I really like acronyms for remembering lots of different things because I find it really hard to remember this off by heart. So in the exam, if you forget it, it's Mr. Sheng. And then you just fill in the acronym and then there's all your marks. Cool. So if I'm alive, I must do all of these things. Metabolize, reproduce, sense my environment, homeostasis, excrete my waste, take in nutrients, grow. I am alive. Yeah, Olive's put she learnt Mrs. Nerg as well. I learnt Mrs. Nerg as well. Right, the next thing we need to do is just do a couple of definitions before we can move on. So definitions is always a thing in biology, right? And it will be in the exam, like for 1 mark, give me the definition of this. So this is from small to biggest, okay? So who can tell me the definition of a cell? If you had to define, Sernan's put small snack. If you had to define a cell, how would you define a cell? Hit me up. What is a cell? What is a cell? Ms91 has put tiny. Can confirm cell is tiny. Okay? But if you had to give like a 1-sentence definition of a cell, what would it be? Smallest component of the body where several processes happen like respiration. I'm going to give Zenon like half a point, yeah? So normally people say it is the basic unit of life, okay? That's what I would define as a cell. Basic unit of life, okay? Now, when multiple cells work together to perform a function, that's a tissue. So all you would write is multiple cells working together to perform a function. And I've got a little example on the right-hand side. Okay, so this is a heart cell, okay? And all of the heart cells work together to make up cardiac tissue, okay? The cardiac tissue is a special tissue that makes up your heart, right? So then if we go down one, well, if we say, oh, well, what's an organ? An organ is literally just, oh, where's my text? An organ, text! You won't let me do text, guys. So an organ is a group of tissues working together to perform a similar function. You can see where I'm going with this. So a group of tissues working together to perform a function. So if we're going down one, what do we think an organ system is? If we're doing basically the same thing but going down, what do we reckon an organ system is? On the chat. Yeah, exactly. A group of organs working together to perform a similar function. So these definitions are really easy, but they are quite important in biology. So I'm going to put group of organs working together to perform a similar function. And an organism is just all of the organ systems working together. So what's Leonardo put? Tissues make organs, organs make systems, systems make individuals. I like that. It's a nice little rhyme. So all you have to know in biology is that the cells work together to make a tissue. The tissues work together to make the organ, the organ system, the organism, okay? And do you guys know how many organ systems there are? Can you give me an example? And I've given you a clue here with the picture. So how many organ systems are there? I think it is important we know this. Olive's put in capitals, respiratory. Give me a number. Olive's going, digestive. How many? How many organ systems do we have? Catherine's put 11, all right. Let's name them. So organ systems. Respiratory. We've got digestive. This is going to be a really messy slide, sorry guys. We've got skeletal, good. We've got nervous, nervous system. We've got, keep going guys, we've got loads more to do. How many more organ systems have we got? On the chat. Immune. Good. Someone's put cardiovascular, but I'm going to give you half a point and give you like, well, I call it circulatory system, but we'll go cardiovascular. Right, in the chat, how many more have we got? So we've got skeletal. Alice Stone has put skeletal muscular. Muscular is actually a separate one. Okay, muscular is a separate one. We've got nervous, we've got skeletal. You've also said, okay, we're missing to do with we. Okay, to do with we, please. Your organ system to do with your we. That's a big one we're missing. What do you think it's called? My we system is my, I'll give you the first letter in the chat. System to do with my wheeze. Urinary. Okay, I've got my urinary system. Oh, someone's put renal, yeah. Urinary system. What about hormones? We need a hormone system. So your hormone system is your endocrine system. And we've got a couple more. What about making babies? Okay, so we've got male and female reproductive. I can't fit reproductive on my slide. Male and female reproductive. We've also got two more. Okay, two more. Lymphatic, which is kind of like your immune system, but it drains all of the fluid from your cells all the time. And we've also got, I'll pop it up here, your skin. Okay, your skin is an organ system, and I love this word. It's called your integumentary system. That's your skin, okay? Your skin is a whole organ system because it's made up of like hair follicles, sweat glands, all sorts of different cells. So your skin is called your integumentary system, okay? Integumentary system. Because it's made up of your skin, hair, nails, etc. So it's all those things together. Right, we took a bit of a tangent. We need to get back on track. So, if we look at all life on Earth can be split into what's called domains. So here I've got 1, 2, 3 domains of life. And this is based on like the characteristics of the organism. So whether they're a single cell on its own or their genetic information is loose inside the cell. But we can't do that today. So we've got 3 domains, and they are archaea, bacteria, and eukarya. But what we want to do today is focus on these two things here. So all life on Earth is basically split into two types of cells, prokaryotes and eukaryotes. Now, you might have heard of eukaryotes before. Eu means like good or true, and karyon means nucleus. So eukaryotes always have a good nucleus. They have a nucleus, okay? And one of the big things you need to do for A-level is to be able to draw and name and describe every single thing inside a eukaryotic cell. So now we're going to draw one together from scratch, okay? So this is my basic blob outline of a cell. And normally at GCSE, all you do is do nucleus, cytoplasm, okay? But we're going to do a little bit more complicated than that today. So we're going to go through and do the different organelles inside the cell. Now, when I say organelle, I mean anything inside the cell that is bound by a membrane. So it has a membrane. So what things do I need to add to my cell? Number one, what is the biggest thing I need to add to my cell? Probably the most important thing, beginning with N. So what do I need to pop on my empty cell first? Yeah, you guys have all said it, nucleus, right? So I'll pop my nucleus in. What is inside my nucleus? What is inside my nucleus? Please. What is inside my nucleus? I'll draw a little thing here. Oh, this is an awful alpha helix. So inside the nucleus, yep, is genetic material stored in the form of DNA. Okay, genetic info, DNA. What do I need to pop in my cell next? What else? So yes, I'll pop cytoplasm up here. Cytoplasm. What else does my cell need? And we're not doing DNA or chromosomes, we're just doing organelles. Okay, so bigger than DNA. So what else does my cell need? What else am I missing? Okay, what's inside a cell? A eukaryotic animal cell, I should say, coming from the comments. This is an animal cell, not a plant cell. We are not doing plants. Right, Alice Stone's put ribosomes. I will give you ribosomes, but what structure are they found on, okay? Yes, we do have some ribosomes kind of free in the cytoplasm, but they're connected to a much bigger structure. So no, we're not doing chloroplasts, we're just doing an animal cell today. So what structure, not doing chromosomes, we are doing this structure that comes off the nucleus here. Ooh. Oh no, my pen. I'll try and place it a lot lighter. Right, so what is the structure that comes off the nucleus called? And it's made up of these like tubules. Do you guys recognize this? This structure comes off of the nucleus and it's covered in ribosomes. What is this? It's an organelle that is attached to the nucleus and it's got lots of ribosomes on it. What is this organelle? Do you guys know? Yeah, Alice Stone's put ER, but what type of ER? So this organelle is called the rough endoplasmic reticulum, or RER for short, okay? Now, we're going to do what everything does in a minute. We just want to stick it on our diagram of the cell, okay? So this here is the rough endoplasmic reticulum, or RER for short, and it's covered in ribosomes, okay? We'll also pop ribosomes here. Ribosome. What else do I need in my cell? What else am I missing? There's a big old space here. We've already got ribosomes, Olive. What other organelle do I need? I will give you a clue. I am the powerhouse of the cell. What am I? I am the powerhouse of the cell. Mitochondria. Yes, we need a mitochondria. Powerhouse of the cell. Mitochondria. I'm going to draw him like this. Going to do loads, because we've got loads. Mitochondria. Cool. Mitochondria. Pop him in. What else do we need in my cell? My cell's looking very empty right now, guys. What else do I need in my cell? What other organelles do we think? That you know of. I'll give you a second. Okay, I'm going to give you a clue now. So the next organelle that you need to know for A-level, I always draw it looking a little bit like a Wi-Fi signal. Like this. This is called, does anybody know what this is called? This Wi-Fi looking organelle. What do we reckon this is? Alice has put the smooth endoplasmic reticulum. No. Let me get my text. Ooh. Yes, Olive's right. This is the Golgi. So some people call it the Golgi body, or some people call it the Golgi apparatus. Okay? Same thing, right? This is the Golgi apparatus, or Golgi body. We're going to learn about what the Golgi does in a minute. And Alice Stone has put smooth endoplasmic reticulum. We need a smooth. So the smooth endoplasmic reticulum is a similar structure, my pen, is a similar structure to the rough endoplasmic reticulum, but it does not have ribosomes on it. That's why it is smooth, okay? Smooth endoplasmic retic, S-E-R for short, okay? Cool. So we've got our rough endoplasmic reticulum that's covered in ribosomes. We've got our smooth endoplasmic reticulum that is not covered in ribosomes. We've got our Golgi. We've got our mitochondria. We've got our nucleus. What else am I missing in this cell? Okay, I've got a few more things to go. Few more things that I'm missing. Yes, Olive, lysosomes. Okay, we need to pop some little lysosomes in. We'll talk about them in a minute. Pop some lysosomes in. What else am I missing that you guys know are inside cells? Leonardo, you've put ribosomes are rough makers. What do you mean rough makers? I'm going to give you a clue. It's to transport things around the cell. So I need something here to transport everything around the cell. Kind of like a lysosome, but not quite. Begins with a V. Beginning with a V in my cell. What is it? They are vesicles. Okay? Vesicles. So I'm going to pop them there. So these are vesicles. And then I'll also pop I want my lysosome label here. Lysosome. Okay? So, obviously we've got our membrane, but we don't do membranes today. There is one other thing that you need to know for A-level that might be new, okay? And I'll just tell you the answer. And this is what's called a peroxisome. And I'll pop it on the board here. Peroxisome. This is another, like, tiny little bubble inside the cell that's going to do stuff, okay? So, these are, oh, one more thing we've forgotten, that moves things around the cell like a road system inside the cell that's made up of lots of stringy fibres that cover the entire of the cell. What is that? One last thing. Covers the entire of the cell like strings that I transport things on. What am I missing? And it's going to ruin my diagram now that looks so pretty. So if I start drawing, I know this looks really ugly. If I start drawing these lines all over my cell, what do you think these are? Okay, I did it in my very first lesson, I think. This is the cytoskeleton, okay? And we'll talk about that in a minute. And I am aware we have spent ages on this cell, so I'm going to move on, okay? Right, So these are all the things inside the cell you need to know for A-level. Now we need to go through and see what they do, okay? What does the nucleus do? Definition of a nucleus, let's go. Definition of a nucleus. What do we think it does? Definition of a nucleus. I'm going to start filling in some of them so we can get through all of the stuff today. Right, definition of a nucleus, yeah. Contains DNA, contains genetic material that encodes for proteins. Remember, DNA encodes for proteins, okay? Leonardo's put like an exoskeleton for the cell. Correct, we'll talk about it in a minute. Plasma membrane is a semi-permeable membrane, i.e. it chooses what goes in and out of the cell, okay? But we're going to do it in a bit more detail next week. The lysosome contains enzymes to degrade things. So anything inside your cell that is like dead, dying, needs recycling, the lysosome will come along, munch it up and just degrade everything and recycle it. Okay? What else have we got going on the chat? Okay, what do we think a vesicle does? Do you know what a vesicle does? A vesicle. Vesicle. While you're thinking of vesicle, I'm going to do peroxisome because it's a new one. So a peroxisome, this is a new thing for A-level. This is a tiny little bubble inside, little organelle inside your cell that is involved in metabolism, and it kind of has lots of enzymes inside that can metabolize like fats for energy. So that's what peroxisomes do. They're involved in metabolism. They can break down fat for energy, okay? A vesicle is literally just a transport bubble, okay? Contains no enzymes. And this is the way that cells transport things inside, okay? So it packages it up into a little vesicle and sends it off around the cell. Next, the Golgi body. Right, Golgi might be a new one. Think of the Golgi as a postage and packaging system. Okay, so what it does is it modifies proteins and then sends them on their way to wherever they need to go. Okay, so it's a modifying system and a packaging system. The rough endoplasmic reticulum, let's pop a definition next to the rough ER. Oh, this contains ribosomes. And what do ribosomes do, guys? This is a really important one. What do ribosomes do? They are the site of... What do ribosomes do? Please tell me. They are the site of protein synthesis, correct? Site of protein synthesis. Smooth ER, no ribosomes. He's involved in making hormones and fats. Smash through these definitions. Mitochondria, powerhouse of the cell. We all know this one. Powerhouse of the cell. What molecule, energy molecule, do mitochondria make, guys? 3 letters. What do mitochondria make? That is the energy of the entire human body. They make... What do they make? 3 letters. Yes, it's the site of respiration, but what does it make? Final product. Yes, ATP. ATP is the energy currency of the cell. And I think you do need to know this for A-level. We're not going to do it today. The actual process of which we make ATP is called oxidative phosphorylation. For real. Whoa! Where's my bloody... Oh, my label's gone. Okay, well, we make ATP. Right. Next one we've got to do. Ribosome. Makes proteins. Oh, what is going on with my... Right. Makes proteins. But the key word we want to know is translates proteins. So it translates an mRNA code into a polypeptide chain. Remember, a polypeptide chain is a protein, okay? Right, we'll skip cytoplasm. Just so you know what a cytoskeleton looks like, this is an image I showed from one of my really first lessons. See all the green strandy filaments? These are basically filaments that cover the entirety of the cell, and they help to transport things. So if I want to move a lysosome from this end of the cell to this end of the cell, I don't just push it out into the cytoplasm, it doesn't float away. I have to send it down this filament of the cytoskeleton, yeah, like a road. I don't just swim around in the cytoplasm. Okay, so A-level, you also need to be able to identify animal cells from photos. So what do we think number 2 is? What do we think number 2 is? What organelle does number 2 look like? And we'll pop it on. What is number 2? Talitha's put the RER. No. What do we think number 2 is? Yeah, this is the Golgi. So this is a microscope image, a high-powered microscope image of a Golgi. Just doing a time check. What do we think number 2 is? And it looks very different in this image compared to what we've drawn it. What's number 3? What is number 3? Leah's put the internet sign, yeah. What is number 3? Someone's already said this. Number 3 is the RER. So this is what the RER actually looks like. It's basically loads of tubes all folded on each other that all contain ribosomes. And number 4 is obviously, what's number 4? Number 4. What is number 4? Otherwise, I'm just going to tell you the answer. Yes, mitochondria. Number 4 is the mitochondria. And number 1 is the entire cell. Okay, so this is what an animal cell looks like under a super high-powered microscope. Okay, we're going to move on. 2 definitions I would like to tell you today before we move on is transcription and translation. Who knows what transcription is? What is transcription, if you've ever heard of it? What is transcription? These are 2 super important processes. We're telling each other ATP jokes. What is transcription? Definition. What is it? Alison's put copying the DNA code, half a point, but what am I copying it into? Yeah, so I'm going to put DNA to, what am I copying it to? RNA, yep. So transcription is making DNA to RNA. Translation is mRNA to what? mRNA to what? mRNA to... I'll wait for you guys to tell me. No, not DNA. Translation is mRNA to protein. Because remember, the whole point of the cell is he wants to make proteins, okay? So transcription is I am transcribing, I'm like a scribe, the DNA to RNA. Translation, I am translating the RNA message into a protein. And just in case someone asks me, we don't directly transcribe DNA to our mRNA, okay? We transcribe it to what's called pre-mRNA. There's like an extra step that happens in between RNA and mRNA, okay? So there's an extra step that we'll do one day. Cool. Prokaryotes, right? Same thing. Karyon means nucleus. What? And pro means before. So prokaryotes do not have a nucleus, or they lack a membrane-bound nucleus, okay? Oh, my pen, they do not have one, okay? So we're going to quickly smash through prokaryotes, okay? Now, there are different types of prokaryotes, and you might recognize the ones like bacteria. So these are ones like E. coli and cholera and all the things that give you diseases. But we also have another class of prokaryotes called archaea bacteria. And these are ones that are found in like deep water thermal vents or in these like acidic sulfur lakes, right? And I love these. These are called extremophiles. Extremophile. Sorry, there's a bit of a lag on my pen. Extremophile, which means they love extremes of temperatures or extremes of anything. So you can see here, like one bacteria that love high temperature, they are called a thermophile for temperature. Ones that live in these like acid lakes are called an acidophile. Ones that love lots of salt are called a halophile, okay? So these are all bacteria that love extreme conditions. And fun fact, I use one in the lab every day, okay? So this is an extremophile called Thermus aquaticus, okay? And he lives in these hot springs, these hot thermal vents. And what scientists have done is they've extracted a special enzyme called TAC, T-A-Q, polymerase. I use TAC polymerase every day in the lab in this machine to do a PCR so I can find out how much of a gene a cancer cell makes. Boom. Okay? So we have this special enzyme that we use every day in the lab that we've got from these thermophilic bacteria, which is really cool. Right. What's in a bacteria? I'm going to just do a time check. Okay, we can do this. Right. So bacteria do not have a membrane-bound organelle. Olive put how big are the machines? Like the size of a microwave, Olive. So bacteria do not have a nucleus. Okay, they've just got one big old chromosome. No membrane, no nucleus, no nothing. They've also got, someone's got it, plasmids. Plasmids are circular, piece of circular DNA. Circular DNA. Then we've got our one big chromosome. No nucleus. No nuclear membrane, we'll put. What else have we got, guys? Let's go, go, go. And I'll keep up. Yes, flagella. Put me flagella on. Flagella. Flagella. What else have we got? We do have some ribosomes. Okay, we'll pop some ribosomes in because we still need to make proteins. Ribosomes. What else have we got? This is nice bacteria. Pili, yes. Put me pili on. Pili, 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 pili. And one other thing. What do we need? What do we need? Something that goes around the outside of the bacteria. I pop it in yellow. All the way around the outside. What else have we got that you can remember? This is such a badly drawn bacteria, soz. Someone's put wall. It does have a cell wall. But we need one other thing that's made of slime. So yes, we've got a cell wall. And we've got one other... Thank you, Miz. It is a work of art. We've also got a slime capsule. Remember, bacteria have slime capsules. Yeah, like a slimy coating. Cool. Look at this beautiful bacterial cell. So these are all the things we've got to put in our bacteria. Right. Let's smash through. Sorry, Leonardo, it does have a membrane. Okay, let's do a triple layer. So we've got cell membrane, cell wall, slime capsule. That's the order it goes in. So it goes cell membrane, cell wall, slime capsule. And this is from inside to outside, okay? Membrane, wall, capsule, 3 layers. Done. The cell wall, we won't do all of these definitions because we are running out of time. Olive's put they are very well protected, yeah. So what does the cell wall do? We're not doing cell membrane because that's for next week. So the cell wall, it kind of makes it rigid, gives it structure. The slime capsule stops the bacteria from drying out and helps him to stick to surfaces. Okay? Remember, you can always go back and rewatch this, but I'm just conscious there's another lesson on at 11, so we're going to try and get through these definitions. Flagella, swimming, movement. Pili are the little hair-like structures that help them stick to things, okay? Oh my God. Boom, prokaryotic cells, done. Plasmids. What are plasmids for? This is what I wanted to spend some time on. What is a plasmid for? I've run a little bit over, sorry. Alison's put genetic rings they can share. Yes, I'll give you half a point. So bacteria's, yes, and they pass them round. So, and what example are they particularly pertinent to? Okay, so let's say I've got some antibiotic that I add to my bacteria. Ooh. I've got some antibiotic and it infects this bacteria, well, it kills this bacteria. Then what do the plasmids do, or what can the plasmids do? Antibiotic resistance, correct. So these plasmids, the bacteria, can put the gene for antibiotic resistance. I'm just going to call it AR, antibiotic resistance, and it can pass it to its other bacteria friend and tell him, hey, there's antibiotic coming. Make sure you take this gene from me and become resistant. I'm going to put here, take my gene for protection. Okay? Yes. And it does this through a process called horizontal gene transfer. Clue is in the name. I'm just passing my gene over to my friend. Okay? So, in conclusion, bacteria use plasmids via horizontal gene transfer to give other genes that might confer protection or in times of stress to their other bacteria friends, okay? That's all plasmids do. Whoa, we've got time. What is the difference between these two bacteria? Number 1 and number 2. What's the difference? What do you think the difference is between bacteria 1 and bacteria 2? Don't say pink and purple. What's the difference? Zenon's put the second one's a bit smaller. Perhaps. What do we think is a difference? Leonardo's shrugging. Okay. One thing you need to know about bacteria is they can all be split up into 2 groups. And this is called, you might have heard this, gram-positive and gram-negative. Okay? Bacteria can be split into 2 groups, gram-positive and gram-negative. And this is based on whether or not they have a thick sugar inside their cell wall. So we're going to look at that now. So what I do is I take my bacteria and I add this stain to them. This is a special stain called crystal violet, and it is a bright purple stain, okay? If the bacteria has this thick sugar in its cell wall called peptidoglycan, key word, peptidoglycan. This is a big sugar molecule. Peptido, my pen is slow, glycan, okay? Thick sugar. If my bacteria has this thick sugar in its cell wall, I will soak up all of the dye, okay? And I'll become bright purple, yeah? So this would be a gram-positive bacteria because I'm positive for the purple, I'm positive for the peptidoglycan. The opposite happens with the gram-negative bacteria. I don't have a thick wall of peptidoglycan. I am actually protected by an outer membrane, so the dye, it cannot get in, and I do not go purple, okay? So that is the difference between gram-positive and gram-negative. And it's really important in the context of antibiotics, what antibiotics we use. So for some antibiotics, they only work against gram-positive bacteria. Some antibiotics only work against gram-negative bacteria. So we have to know what bacteria we're working with first before we can give the antibiotics to that person, okay? So this is called gram staining, and it stains for the peptidoglycan sugar in the cell wall of the bacteria. I've done so much talking. Right, we don't have time for that, guys. How do prokaryotic and eukaryotic cells divide will lead into our next lesson, okay? Eukaryotic cells divide by mitosis. Prokaryotic cells divide by binary fission. And we will cover this next lesson, okay? And we'll also go back and finish our compare and contrast. Whoa, oh my God, so we've done loads today. Right, I've got 3 minutes left. Today, we have done the structure and function of prokaryotic and eukaryotic cells. Bless me. We have not compared and contrasted them, I'm sorry. I ran out of time. We did the structure and function of organelles. We are going to do mitosis next week. We're going to do membranes next week, and we're going to do diffusion and osmosis. No, we're not. We're going to do them on Friday, okay? This is Friday's lesson. So we're going to crack on with mitosis. We're going to do membranes. We're going to do osmosis and diffusion. Remember, you can rewatch this lesson anytime you want because we have just crammed about 3-4 weeks worth of A-level biology into 1 hour. And now I'm going to go and have a drink of water. Okay. Goodness. My breath. Right. I hope you've found this very crash course useful. Next week, mitosis. Friday, mitosis, membranes, and diffusion and osmosis. Remember to use the mark scheme for your revision. Oh my gosh, and I will see you on Friday, okay? Hopefully you've enjoyed this like super crash course, okay? I know we can't go through everything in that much detail, I'm sorry. But hopefully it's helped teach you some of like the new words you're going to encounter in A-level and how much detail you need to go into in A-level as well. Leonardo's put, am I in the lab? I am in the lab right now. I've got my little key card on for work, so I'm going to go and do some experiments now. So I will see you guys on Friday. Goodbye. Ciao, ciao.